Hello, everybody, and welcome to today's class on intuitive statistics. We just want to take a brief bit of time and not go over all the statistics that are possible and things, but why we need to think about statistics and how it will apply for our class moving forward. So we've generated all this data, we've clustered it, things are looking good, the biology seems to be coming out of it, but moving forward, we're going to want to develop a statistic to say, is this DNA binding protein significantly bound to promoters versus other places in the genome? Does it overlap with transposable elements more than is expected by chance? And so we're going to walk through some of the basic statistics and just get an intuitive feel of why we're going to use this one statistical framework that will work for all of our data, which is essentially generating an empirical null distribution where we randomize the actual data and ask, is this feature significantly overlapping at random compared to what we observed in the actual experiment? So we'll get to that in a second, but first we need to start with the p-value. And we all have heard of p-values, and usually we always assume if it's 0.05 or less, it's significant. There is no real cutoff here with p-values. But that's kind of the standard that we use. And that sounds good until you start making a lot of observations. So in the human genome or gen code, we're going to be looking at some cases up to 60,000 features. So 5%, the p-value of 0.05 means about 5% of our data, our significant values are probably wrong. So 5% doesn't sound that bad until you start adding up multiple observations. And you really need to correct for how many observations you're making here. So let's say we're looking at 60,000 genes roughly, then we can expect 3,000 false positives in our data. And that's not good. So this is where p-values really break down in genomic data. If you've done like a few experiments and you're comparing them, that might be more appropriate, but you really need to take into account the number of observations. And in this class, we're making a lot of observations, which means we're gonna have a lot of errors. And 3,000 genes, that's a lot. I mean, that could influence the data in a completely different direction from what the actual biology is. So we don't wanna use just the simple p-value of 0.05. We will use a p-value, but we're gonna use it in a way that's been corrected for or empirically derived. And we'll get to that in a second. Okay, so what we really need is a null distribution. Is what do observations look like at by chance, or what would we expect an observation to look like? And then what is our actual observed observation? And when we have a null distribution or an expected versus observed, we can do a test statistic. We'll talk about the Fisher exact test. Is probably what we'll be using almost for the whole class. And then we're going to get a p-value, but then we're going to correct the p-value because you can get p-values that are significant, but we'll see in a second how we might want to correct that to make sure we're limiting the number of false positives that we're going to get. And a false positive in this case means that we're rejecting something that was actually true. I know it's a little counterintuitive, but it's a type 1 error, and we'll talk about that next. So basically what we'll do is we'll, in the class, derive this null distribution from the actual data, where we permute the data multiple times and just ask, well, what are the chances of seeing this by random? And then what is our observed value? We use those two to create a test statistic, either z-score or Fisher exact test, and then we'll correct those p-values based on the number of observations and distribution of p-values. Okay, so what does that look like? Well, just for some background, what we're talking about mostly today is type 1 errors, or what are called false discoveries. And so what that is that we're rejecting something that was actually true. So we don't want to do that. And we'll look at how that's corrected for. But really what we need to always take into consideration in statistics is how many observations are we looking at? So where M is a hypothesis test. So if we have thousands of hypotheses, M is the total number of those tested, and we can think of each gene as a hypothesis or promoter or transposable element that we're looking for or who we're going to look at moving forward. So we really have to pay attention to this number of observations. And so this is what a typical data set would look like. Here on the x-axis is the p-value in your data. So let's say we had 60,000 genes. This would be the distribution. The y-axis here would be the frequency of seeing this p-value. And what we can see is that these p-values are sort of evenly distributed along the bottom here. And if we draw a line where this dashed line is here, we can see that all the area under that line are not really meaningful p-values. They're almost like p-values that could have occurred by chance, or they're normally distributed, is another way to think about that. So what we can simply do is shift this line up here, and where the real problem comes in is here, that under this line, if we don't correct for this, all of these are going to be considered significant, but they're not actually, they could have come by chance as well. So what we need to do is shift the line up to get rid of these p-values, this number of observations that we said, oh yeah, they're true because of this p-value. We want to shrink that, and this would be a false discovery rate. So we're going to limit the proportion of mistakes that we're making by correcting for this region here. Sort of putting that into words, there's two ways we can do this. Is a family-wise error rate correction. And what that's going to do is making sure you never reject a true hypothesis. So it's going to try and prevent type 1 errors, period. Whereas a false discovery rate is probably more common and is going to say, this is the proportion of your p-values that are probably not true. And then we can correct for that and help make sure that all the observations above this line are more likely to be actually true. So we want to account for the dependency of the structures of p-values. So this is just one example, but we could see that most p-values could be up here, and really that the chances of seeing anything significant in our data was almost zero because the data was basically the same between two samples. But because of the number of observations, there was some false discovery in there. But that can be limited with these corrections. So that's sort of the last step we're going to do. But then we also need to do the test statistic to get the p-values and talk about how we're going to drive these null distributions. So the way we're going to do this in class is we have 460, we reduced it to 438 chip data sets. And each of those chip data sets is different. Some have 1,000 peaks, some have 1,000,000 peaks or 100,000 peaks. Some peaks are small, some peaks are large. Some peaks might come from a bias of chromosomes. So we want to account for those things in the data and saying, okay, so we have 10,000 chip peaks. What would happen if we just threw those around randomly? And so each permutation would be saying, taking the same properties of our data and permuting them. So that's what we're going to focus on in class, is a permutation, where we take the attributes of a given DNA binding protein, we throw it on the genome, and we say, how many times did it overlap? We do that again, we do that again, we do that again. And if we do it 1,000 times, we're going to have the ability to get a p-value of up to 0.001. If we do it 1,000,000 times, we'll get deeper p-values. So permutations are going to be based, the p-values limitations are going to be based on the number of times you resample your data. So why does this matter? Well, we sort of just covered that, is that each DNA binding protein has very different null distribution. So we don't want to just assume a normal distribution, a Gaussian distribution, a Poisson distribution. If the data, and a lot of genomic data does follow the Poisson distribution, which we won't discuss today, but will come up maybe later in the class, is these are known distributions where you can say, okay, well, I assume my data is following a Poisson distribution or a Gaussian distribution, and then you can do a test statistic of observed versus that expected distribution. But for our DNA binding proteins, we can't assume that it is following any type of normal distribution. And that each DNA binding protein is going to have its own sort of distribution of data because the number of peaks, the sizes of the peaks, and the bias in which chromosomes they like to bind to. So we have to account for those things. And it's always best if you can do an empirical null, where you can say, here are some variables in my data that I want to account for. And I'm going to do random samplings over and over and over again and count the overlaps each time, so that I can account for these properties, not biasing my data to being significant when it really shouldn't be. And so this is really the framework we're going to be using, is creating a null distribution empirically, running a test statistic, and then correcting those statistics after that with an FDR. And so this is what I was talking about, where there are normal distributions. So you can do a t-test, or t-distribution is shown here, where whereas we talked about is like k is the number of degrees of freedom, or sort of the number of observations you're making, or variables in the observations, I should say, that you're, excuse me. And so this is a t-distribution here, where I was talking about that distributions are just sort of known in biology, the normal distribution, the t-distribution here, where you have the number of degrees of freedom, which means sort of the number of observation variables you have. And the more observation variables you have, the harder it is to get a significant p-value. Again, correcting for the more observations, the more likely you are to have things by accident. But this is not what we're going to use, because these are assumed distributions that occur often in biology. But again, we want to create our own empirical null that represents our data, specifically each DNA binding protein's data. Okay, so the actual tests we're going to be using is null distributions via permutations. We'll get into how to code that in class. It's kind of cool that you can take your data, figure out its properties, and then use those properties to permute, and then ask what your observed is within a Fisher exact test. What's my observed versus expected of the distribution? And we'll do some false discovery rate. Bonferroni is probably one of the most brutal ways to correct your hypotheses, where you basically are multiplying your p-value times the number of observations. In our case, because we're doing the number of observations by permuting, Bonferroni is probably not going to help us much. So what two types of things we're going to do in class is a z-score and a Fisher exact test. A z-score also gives you a p-value, and a z-score you can think of just as how extreme are you on this distribution. So this is a normal distribution shown here. Our empirical distribution might look something like this, but maybe one side of this tail is longer, and these curves can be pulled and shaped differently, and that's why we're going to empirically determine it. But for this example, let's just say this is our empirical null distribution. And we have an observation here where we say, this is what we observed versus our expected overlap frequency in our permutations. And so if we were here, we would say, okay, we're about, what we need for this equation essentially is the observed, the mean of our null distribution, and then the standard deviation, or how wide this curve is. And really we're, for a z-score, sort of calculating where you sit on the edges of this distribution. So this looks pretty significant. If you just said, oh, I'm 2 standard deviations from the mean, that's pretty good. That sounds, pretty significant, but actually the p-value would be about 0.1 and wouldn't be significant. Now, if you were 3 standard deviations in this distribution, you'd be here, and the p-value would be about 0.0013. So there's lookup tables for where you are, and can be calculated. A z-score can be calculated very easily from an empirical null distribution, because you're just saying, here's my observed value, here's the mean of my empirically permuted data, mean number of overlaps. Let's say you saw a DNA binding protein overlap a promoter 1,000 times, and in the null distribution, it only happened 5 or 6 times. And then what's the standard deviation of that? And then you can get a z-score. And a p-value for that. And we'll be using z-scores quite a bit, because we're driving empirical nulls that may not look as nice as this, but we can get these values from our empirical null distributions. The Fisher exact test is kind of the same idea, except you're sort of calculating the area under the curve of other possible observations. So let's say you observed something here, and this is your empirical null that you did permutations and counted the number of overlaps. And you say, okay, well, I'm here on the distribution. This is the 0.05 cutoff, which means that only 0.05% or 5% of the data is under the area under this curve, meaning that these are very unlikely observations. In a two-tailed test, these are also can be on this side of the distribution. And so what you're really just simply doing is calculating the area under the curve and getting your p-value of what percentage of the data could have been explained by chance. And so in this case, you have a 5% probability of this being somewhere in this random null distribution. And that's where we'll sort of draw a statistical threshold there. And so what we've just talked about here is how to get our null distribution and accounting for the dependencies of the structure of the p-values is also going to be done with false discovery rate corrections. Our test statistics will be a Fisher exact or a z-score, just basically asking how far away are we from these random possible chances. And so let's take a practical example of this, how we're going to move forward for now, and we'll introduce some other statistical things later. But really, this is a logical framework that works for almost any data, and is a very, I think, clean way of approaching your statistics. So just to summarize all that we've talked about, it really gets down to something pretty simple. Is we have 438 different DNA binding proteins, all with different sort of properties. So let's just assume our DBPx has 10,000 peaks, and these peaks range from about 200 to 700 base pairs, and that there's a bias on chromosome 1. Why would there be a bias on chromosome 1? Why would you have more peaks on chromosome 1? Well, it turns out chromosome 1 is the biggest chromosome in the human genome. So because it's bigger, you're more likely to get a binding event by chance. So what we're going to control for is the number of peaks, because each DNA binding protein has very different amounts of peaks. They also have different window sizes. Some cover large windows, and we need to be sure that when we take this data and we go put it randomly in places in the genome, that we're representing the structure of the data as well. And then we can count how many events were on each chromosome, what the window size was for that chromosome, and the peaks for that chromosome. And then what we can do is do permutations on every single chromosome. So I think that's kind of confusing, but really what we're saying is we're going to represent the data as it is, and then sprinkle it randomly around the genome and ask how many times did we see an overlap with a promoter? Like, does this DNA binding protein bind to promoters significantly more than we'd expect by chance? Same with a transposon. That's an example I'll show in a second that we found last year that was quite interesting. There's so many transposons or repeat elements in the genome that when you do these permutations, you get a lot of overlaps by chance. So finding a DNA binding protein that prefers a transposon over other regions of the genome was very difficult to find, and we really only found one interesting one. And that's because exactly why we did this is that there's so many of those events that if we didn't correct for the number of those events by chance, then we would be using sort of a bad statistical framework. So once we do that, we're going to take these properties, and for each permutation, we're going to throw them down on the genome and say, how many times did I see overlap with a promoter? So the more number of peaks we have, the larger the peak window size, the more likely we are to get an overlap by chance, just randomly by scrambling the data and saying, put it wherever you want, and then count them, do that again, do it again, do it again. And each time we count, we'll get a null distribution that will come out of that. And that'll be our expected data or our empirical null distribution. But we still have our original data where we derived this sort of set of properties to control for our statistics. And we'll use our observed value of how many overlaps we saw in our actual data versus the random data and use a Fisher exact test or a z-score. And then we'll know if the result is significant or not. And these are p-values you can kind of care about because they've been well crafted to control for biases in the data, obvious biases that would make overlaps happen by chance and not be true significant biology. And so the example of that is sort of what you can do is you can say, okay, let's say on the x-axis here, we have the number of overlaps with a promoter or transposable element. And this is for one DNA binding protein, and this is the frequency that we saw this. So let's say we did 1,000 permutations, and we saw that 10 times we see 33 overlaps, about 25 times we see 34 overlaps. You can see how this almost looks just like that normal distribution, but what we're accounting for is this differences in structures here. So these are much lower. So 35 turns out to be less frequent than 34, but 36 is more frequent than 34. So there's some flux here or variance. And that's where jackknifing, which we're not going to use, can come into play to take considerations that, hey, look how far off you are from a normal distribution. We should maybe not consider this data point as much as the others. But okay, so this is simply just deriving this empirical null by saying it overlapped 39 times very infrequently, twice. And let's say our observed value was 47. So this never occurred in our permutations, and we can see that if we drew sort of a line, we'd be at the very extreme tail of this distribution's projection, and this would be highly significant. And so we can also do this as a z-score, where we say, here's the mean of our empirical null, our standard deviation of it, and our observed value here. And that's what we did in class. And what you can see here, the z-score of 100 is very good. It means you're at the very extreme of the empirical distribution. And so what we saw was this SVA transposon, where when we threw the data around 1,000 times, the observed value was highly significant, sort of like the example we just saw here. The observed was actually never found in the random sampling. And it turned out 3 proteins preferred this SVA element over anywhere else in the genome. And that's what we were testing with our empirical null. And we saw these 3 proteins. We won't go into it. We talked about it already in class and in the paper. But that's why that's important. And really what we saw for most things is they're either enriched or depleted for everything because there's so many of these events. But this one was very unique, where 3 proteins prefer to bind to this transposon, which is the newest transposon in the human genome. So it's very human specific. So that was an interesting thing. And what we're going to be doing, I think, in 2 classes from now, is making random permutations for each DNA binding protein with all of these repeats, and then asking which DNA binding proteins, if any, had significant, where it preferred this over random places in the genome. So with that, we will continue moving forward and start developing null distributions and checking properties of our DNA binding proteins, of whether they prefer to bind promoters, whether they prefer to bind enhancers, transposable elements. And we'll sort of walk through several different genomic features where we make random distributions, count the number of times those features overlap by chance, and then what we saw when we in our actual data. So with that, thank you, and we will see you soon.